Welcome you all to in today's lecture. In continuation to the previous lecture, today we will discuss the alternative pathway of ammonia assimilation. Earlier theories proposed that reductive amination played a central role in the assimilation of ammonia. The enzyme responsible for this process is glutamate dehydrogenase, which facilitates the conversion of ammonia in the form of ammonium ions and alpha ketoglutarate into the glutamate. So, this reaction is basically a reversible reaction. In plants, these are two distinct types of GDH. One type known as NADH dependent GDH, which operates in mitochondria and relies on NADH as the reducing agent for the reaction. The other type is NADPH dependent GDH, which is located in the chloroplast. This information highlights the compartmentalization nature of ammonia assimilation in plant cells with different GDH type operating in a specific cellular organelles. This figure shows that GDH catalyzes both reductive amination, in which 2-oxoglutarate is converted to glutamate, and oxidative deamination, in which glutamate is deaminated to generate 2-oxoglutarate. In the process of reductive amination, the reaction involves the simultaneous oxidation of NADH to NAD plus or NADPH to NADP plus. In this context, the reducing agent, that is NADH or NADPH, donates electrons to the reaction, facilitating the conversion of ammonia and a ketone or aldehyde into an amine with the concomitant formation of NAD plus or NADP plus. On the other side, during oxidative deamination, the opposite occurs. The reaction leads to the removal of amino groups from amino acids, producing ammonia and keto acids. Importantly, this process results in the generation of reducing power, where NAD plus or NADP plus is reduced to NADH or NADPH. The newly formed reduced coenzyme can then be utilized in various metabolic pathways to provide energy or serve as electron carriers in other redox reactions. However, reductive amination involves the oxidation of NADH or NADPH, while oxidative deamination generates reducing power in the form of NADH oblique NADPH. These processes are integral parts of cellular metabolism, contributing to the dynamic balance of redox reactions within the cell. Moreover, GDH could potentially play a role in assimilating increased amounts of ammonium ions, especially those generated from photorespiration within the mitochondria. The activity of GDH tends to rise in the presence of elevated ammonium concentrations, suggesting a potential function in detoxification. Additionally, GDS might be crucial in the breakdown of glutamate, producing 2-oxoglutarate, which could serve as an intermediate in the tricarboxylic acid cycle for energy generation. This process may be particularly significant in the absence of light during seed germination or in aging leaves. Now we will discuss about transamination reaction. After nitrogen is assimilated into glutamate and glutamine, it becomes integrated into other amino acids through transamination reaction. Aminotransferases, and that is enzyme facilitating these reactions, which play a crucial role. As seen in the figure, the aspartate aminotransferase drives a reaction wherein the amino group from glutamate transferred to the carboxyl group of oxaloacetate. Aspartate, an amino acid, is involved in the process such as malate aspartate shuttle, aiding the transfer of reducing equivalents between the mitochondrion, chloroplast and cytosol. Additionally, it participates in the transportation of carbon from the mesophyll to the bundle sheath for C4 carbon fixation. It's important to note that all transamination reactions require pyridoxal phosphate, a form of vitamin B6, as a cofactor. Aminotransferases are distributed across various cellular compartments, including the cytoplasm, chloroplast, mitochondria, glyoxysome and peroxisome. Notably, aminotransferases located in chloroplast are likely to play a crucial role in amino acid biosynthesis. This is evident from experiments where plant leaves or isolated chloroplast, when exposed to carbon dioxide labeled with radioactivity, efficiently incorporate the labeled carbon into amino acids such as glutamate, aspartate, alanine, serine and glycine. Asparagine, initially discovered in asparagus, marked the first identification of an amide. Beyond its role as a protein compartment or protein component, asparagine plays a crucial role in nitrogen transport and storage due to its stability and favorable nitrogen to carbon ratio, specifically 2n to 4 carbon for asparagine as opposed to 2n to 5 carbon for glutamine and 1n to 5 carbon for glutamate. The primary route for synthesizing asparagine involves the transfer of amide nitrogen from glutamine to aspartate. The asparagine synthetase, the enzyme responsible for catalyzing this process, is basically present in the cytosol of leaves, roots, and nitrogen fixing nodules. In maize roots, especially under conditions of potentially harmful ammonia levels, ammonium may substitute glutamine as the supplier of the amide group. The expression of genes coding for AS and the enzyme activity are inhibited by high levels of light and carbohydrate conditions that typically stimulate plastid glutamine synthetase and ferredoxin dependent glutamate synthase. This opposing regulation of the competing pathways helps to maintain a balance in the carbon and nitrogen metabolism of plants. Conditions characterized by abundant energy, such as high light and carbohydrate levels, promote GS and GOGAT while inhibiting asparagine synthetase. This preference facilitates nitrogen assimilation into compounds like glutamine and glutamate, which are rich in carbon and contribute to the synthesis of new plant material. Conversely, energy limited conditions hinder GS and GOGAT, leading to the stimulation of AS. This shift in favor of AS encourages nitrogen assimilation into asparagine, a compound with a high nitrogen content that is stable enough for long distance transport or prolonged storage. So, this figure basically displays the biosynthesis of the aspartate and asparagine amino acids that are part of the nitrogen metabolism in plants. Aspartate is typically synthesized through the amination of oxaloacetate. This reaction is catalyzed by the enzyme aminotransferase, aspartate aminotransferase. Aspartate is a precursor for several other amino acids. Asparagine is synthesized from aspartate in two-step process. First, aspartate is converted to asparagine in a reaction that is characterized by the asparagine synthetase, which involves the transfer of amide nitrogen from glutamine to aspartate. The amide nitrogen donor in this reaction is glutamine, and this reaction helps in incorporating the nitrogen into asparagine. In terms of regulation, the expression and activity of enzymes involved in these pathways are often regulated by environmental and cellular conditions. For instance, the levels of light, carbohydrates, and nitrogen availability can impact the expression of genes coding for enzymes like asparagine synthetase. So, the biosynthesis of aspartate and asparagine is tightly regulated to balance the carbon and nitrogen metabolism in the plants. Understanding these pathways is crucial for comprehending the physiological and metabolic responses of plants to varying environmental conditions. If you have specific questions or need further details on any aspect, you can feel free to ask. So, from this slide, we can say that indirect evidence suggests that oxaloacetic acid can undergo amination to form aspartate, while pyruvate can be aminated to produce alanine. These processes involve the addition of an amino group to oxaloacetic acids and pyruvate, resulting in the synthesis of aspartate and alanine respectively. Aspartase is an enzyme that has been identified in several species. This enzyme plays a role in catalyzing the reversible amination of fumarate to generate aspartate. Aspartase facilitates the conversion between fumarate and aspartate, providing a means for the incorporation of amino group into compounds. Aspartase catalyzes the reversible reaction where fumarate, a compound in the citric acid cycle, can be aminated to form aspartate. This reversible process allows for the dynamic conversion between fumarate and aspartate, contributing to the metabolic flexibility of the cell. Ammonium ion basically have the capability which can enter organic compounds to form amino acids through several pathways. The reaction involves the conversion of 2-oxoglutaric acid and ammonium ion into glutamate, and the next is the glutamine synthetase glutamate synthase pathway. In this pathway, glutamine synthetase incorporates NH4 ion into glutamate and subsequently glutamate is converted to other amino acids. Another is transamination reaction, where amino groups can be transferred between amino acids through transamination reaction, allowing NH4 ion to be integrated into various amino acids. Amination of keto acids such as pyruvate can undergo amination to form amino acids like alanine. Amination of organic acids including oxaloacetic acid can undergo amination to form aspartate, as well as the transformation of fumaric acid and ammonium ion into aspartate. Among these processes, the amination of 2-oxoglutaric acid is particularly significant. Its primary role is in the deamination of glutamic acid, especially during the redistribution or relocation of nitrogen. Understanding these processes is crucial for comprehending the intricate biochemical pathways involved in amino acid biosynthesis and nitrogen metabolism in living organisms. These mechanisms contribute to the synthesis of essential building blocks for proteins and other crucial cellular compartments. Under condition of abundant energy characterized by high levels of light and carbohydrate, there is a stimulation of glutamine synthetase and ferredoxin dependent GOGAT, while simultaneously inhibiting the asparagine synthase. This combination of regulatory action promotes the assimilation of nitrogen into glutamine and glutamate. These amino acids play a crucial role in the synthesis of new plant materials, contributing to growth and development. Conversely, in conditions where energy is limited, there is an inhibition of GS and GOGAT, along with stimulation of asparagine synthase. This regulatory response favors the assimilation of nitrogen into asparagine. Asparagine, being rich in nitrogen, serves as a compound that is stable enough for long distance transport and long term storage within the plant. In summary, the balance between these pathways is finely tuned based on the energy status of the plant. So, high energy conditions direct the nitrogen assimilation towards glutamine and glutamate for immediate growth and development, while energy limited conditions prioritize the production of nitrogen rich and stable asparagine for long term storage and transport. So, in conclusion, we can say that ammonia assimilation is a fundamental process in the nitrogen metabolism of living organisms. involving the incorporation of ammonia into organic molecules, particularly amino acids. This essential pathway contributes to the synthesis of key biomolecules like proteins and nucleic acids, influencing growth, development, and overall physiological functions. Efficient ammonia assimilation is vital for maintaining nitrogen balance and sustaining essential biological processes, highlighting its significance in the overall metabolism of organism. So, this is all about for today. Thank you.